Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Dr. Rizwan here from the Department of Biochemistry, Independent Medical College, Faisalabad. In our previous lecture, we have discussed the very important topic, that is the apoptosis and the metastasis. And this is the last lecture on the cancer metabolism, and in this lecture we will discuss tumor biomarkers which can be measured in the blood and other body fluids. The biochemical tests are often helpful in the management of the patients suffering from the cancer. For example, the patients with the advanced cancers may have developed the levels of plasma calcium, and the many cancers are associated with the abnormal production of the enzyme, proteins, and the hormones that can be measured in the plasma or the serum. These molecules are known as tumor biomarkers. The significant elevation of some of these biomarkers also occur in a variety of non-cancerous conditions. For example, elevation of the levels of the prostate specific antigen, it is also known as a PSA. It is a glycoprotein synthesized by the prostate cells and occur not only in the patients suffering with the cancer of the prostate gland, but also in the patients with the prostatitis and benign prostatic hyperplasia. In this table, we are seeing the different biomarkers by which we can detect the different associated cancers. The number one is the alpha fetoprotein. It is useful for diagnostic purposes in the hepatocellular carcinoma and the germ cell tumors. And the second one is the calcitonin. It is the diagnostic tumor biomarker for the thyroid or the medullary carcinoma. Next is the CEA, carcinoembryonic antigen. It is useful in the detection of the cancer of the colon, cancer of the lung, cancer of the breast, cancer of the pancreas, and the cancer of the ovaries. The next biomarker is the human chorionic gonadotropin, HCG. It is responsible for the diagnostic criteria of the trophoblastic diseases or the germ cell tumor. The next biomarker is the monoclonal immunoglobulins, and it is a biomarker used for the diagnosis of the different myelomas. And the next is the prostatic specific antigen, and it is useful in the diagnosis of the prostate gland cancer. The elevation of carcinoembryonic antigen, it is found not only in the patients suffering with the various type of the cancers, but also in heavy smokers and the people with ulcerative colitis and cirrhosis of the liver. The fact that the elevation of the tumor biomarkers is usually not specific for the cancer has meant that the measurement of most of them are not used primarily for the diagnostic criteria for the cancer. Now in this slide we will discuss the different mechanisms which are involved in the carcinogenesis. And among the classes of the drugs developed more recently are the inhibitors of the signal transduction, including the tyrosine kinase. The finding of the widespread defects in signaling mechanism in the cancer cells, and in the particular the detection of the mutation in the tyrosine kinase, has led to the development of the inhibitors of these enzymes. The most dramatic success has probably been the introduction of Imatinib, which is used for the treatment of chronic myelocytic leukemia. The Imatinib is an oral administered drug that inhibits the tyrosine kinase formed due to the chromosomal translocation which is involved in the genesis of chronic myeloid leukemia. This drug, Imatinib, it is an ATP analog, competitively binds to the ATP binding pockets of the kinases. This drug has produced complete remission in many patients. It can be combined with other certain drugs, and the other tyrosine kinase inhibitors have also been developed. Two of these are erlotinib and gefitinib, which inhibit the epidermal growth factor receptors. This epidermal growth factor receptors is overexpressed in certain lung and the breast cancers, resulting in the aberrant constitutive signals. In this table, we will see some anti-cancer agents that are based on recent advances in the cancer biology, and the classes of the drugs are inhibitors of the signal transduction, and the drug is Imatinib. It is an inhibitor of tyrosine kinase, and it is used for the treatment of chronic myeloid leukemia. And the second class is the monoclonal antibodies, and it includes the trastuzumab. It is a mAb monoclonal antibodies to HER2 neuroreceptors. These are basically the estrogen proteins and the protein receptors, which is also known as the human epidermal growth factor receptor 2 proteins, and it is used for the treatment of the late stage of breast cancers. The third class of the drug is the anti-angiogenesis agent bevacizumab. It is a mAb to the VEGF. It is vascular endothelial growth factor A, and it is used for the treatment of colon and the breast cancers. The next formulation is the anti-hormonal agents. It is tamoxifen. It is antagonist of the estrogen receptors. It is also used for the treating the breast cancer. The next class is the effect differentiation. It is used in the promyelocytic leukemia, and the effect epigenetic changes. For example, 5-azadeoxycytidine inhibits the DNA methyltransferases. SAHA inhibits the histone deacetylases, and this SAHA is basically the subaryl aniline hydroxylimic acid. It is used in the treatment of a certain leukemias, that is the cutaneous T-cell lymphoma. The other approaches to treating the cancer that are being used or being developed include various types of gene therapies, immunotherapy, oncolytic viruses, inhibitors of the progesterone receptors, aromatases inhibitors, telomerase inhibitors, and application of nanotechnology, for example the nanoshells and the other nanoparticles, the phototherapy, and the drugs that will selectively target the cancer stem cells. So in this table we will see the mechanisms by which the cancer cells can develop the drug resistance. Now we'll discuss the tumor microenvironment role in spreading of the cancer. The tumor microenvironment consists not only the cancers but also a variety of non-cancerous cells and the components of extracellular matrix. The major non-cancer cells present in the tumor microenvironment includes those of the immune cells, that is the T and B lymphocytes, the natural killer cells, macrophages, and mesenchymal cells. The variety of immune cells infiltrate the tumors their anti-tumor effector functions. These are the dampened in the response to the tumor derived signals. The T lymphocytes and the macrophages are reprogrammed. The mesenchymal cells such as myofibroblast and the mesenchymal stem cells are known to facilitate the formation of the cancer stem cells, thus aiding the cancer stem survival and proliferation. And the other mesenchymal cells such as the endothelial cell respond to the paracrine signals in the tumor microenvironment by promoting the endothelial growth factors, leading to the tumor angiogenesis and metastasis. Here are the certain definitions regarding the tumors and the spread of the cancers. These are the adenomatous polyp. It is a benign tumor of epithelial origin that has a potential role to become carcinoma. The adenomas are often polypoid, and a polyp is a growth that protrudes from the mucous membrane. Most are the benign, but some polyps can become malignant. The second terminology is the apoptosis. It is also been discussed in the previous lecture. It is a programmed cell death due to the activation of genetic program that causes the fragmentation of the cellular DNA and other changes. The caspase play a central role in this process. Many positive and the negative regulators affected, and the protein p53 includes apoptosis as a response to the cellular DNA damage. Next is the Burkitt's lymphoma. This is a B-cell lymphoma and endemic in the parts of Africa where it mainly affects the jaw and the facial bones. A reciprocal C-Myc gene on the chromosome 8 and immunoglobulin heavy chain genes on the chromosome 14 is characteristic. The term carcinoma is a malignant growth of epithelial region. A cancer of glandular origin or showing the glandular feature is usually designated as adenocarcinoma. The caspases, these are the proteolytic enzymes that play a central role in apoptosis, but also involved in other processes. Some 15 types of caspases are present in the humans. The leukemias is a variety of malignant diseases in which the various white cells, for example myeloblasts, lymphoblasts, and proliferate in an unrestrained manner. The leukemias may be acute or the chronic. The next term is the lymphoma. It is a group of neoplasm arising in the reticuloendothelial and lymphatic systems, and the major members of the group are Hodgkin and non-Hodgkin lymphomas. The epigenetics refers to the changes in the gene expressions without change of the sequence of bases in the DNA, and the factor causing the epigenetic changes include the methylation of the bases in the DNA, post-translational modification of histones, and chromatin remodeling. The proto-oncogenes are the normal cellular genes which when mutated can give rise to a product that stimulates the growth of a cell, contributing to the development of a cancer. The retinoblastoma, it is a rare tumor of the retina. Mutation of the retinoblastic tumor suppresses a gene which play a role in its development. The patients with the hereditary retinoblastoma have inherited one mutated copy of RB genes and only need a further mutation to develop the tumor. The sarcoma, it is a malignant tumor of mesenchymal in origin, for example from the cells of the extracellular matrix or the other sources. The tumor suppressor genes are the genes whose protein normally restrain cell growth, but when its activity is lost or reduced by mutation, it contributes to the development of cancer cell. So students, here we will end this chapter, and the topics are almost all the covered which are included in your syllabus. So this is very important topic regarding your examination point of view also. So read out it from the Harvard's book also. Okay, Allah Hafiz. Thank you very much. Assalamu alaikum.